As you know, by aging, our brain gradually but undetectable cognitive decline is occurred. And at the molecular level, we will notice that driving aging of the brain include mitochondrial DNA damage, chronic oxidative stress. When you see these two words, right away you understand that you are talking about neuroinflammation, which will lead to oxidative stress. So neuroinflammation, characterized by excessive glial activation. All of us know that this glial activation and overproduction of the pro-inflammatory cytokines and chemokines, which play a role in the pathogenesis for the occurrence of Alzheimer's disease. Alzheimer's disease is the leading cause, as we know, for dementia, all right? And it is characterized by two things, two biochemically distinct types of protein aggregate, which is amyloid beta peptide. Second is aggregated tau protein in the form of intraneural neurofibrillary tangles. Okay, so if you look at this interesting diagram, you will notice that amyloid precursor protein, all right, in the normal condition and in the disease condition. In the normal condition, it will deal with alpha secretaries, will form this soluble form of amyloid protein, all right, and this will be eliminated by ubiquitin proteasome pathway. So what is the problem here? The problem here is that we start to get a different protein misfolded, which lead to aggregate, and this will form like plaque all over the neurons. Of course, there are several risk factors. Most important of them is apolipoprotein A4, okay? But what is the trigger? Why this disease occurs? This is due to the trigger of innate immune mechanism. When you say innate immune mechanism, we are talking about the glial cells, all right? And we will see, okay, what happened to the glial cells? Why they start to do this ugly action? Just to remind everybody that microglial cells in the brain can be activated in response. This is a point. Once you have the protein changed in the structure, it becomes misfolded. Once it is misfolded, it will start to activate the microglial cells. These diverse microglial cells from physiological and beneficial effects to a disease condition. But the question remains, what happens? Why these microglial cells are activated? Just to remind everybody, of course, these are the major part of the innate immune system, the microglial cells, and called as a resident macrophage or the fixed macrophage in the brain, all right? And something very important, look how they are very active. They express all different types of toll-like receptors, i.e. they can detect bacteria, they can detect a virus, and any pathogen in the brain, and this will be activated then why? Okay. Look at here, and this is a very interesting slide. This is the microglial cells in the normal condition, resting condition. They can be activated by any pathogen or any insult, such as injury, stroke, infection, pollutants, neurotoxin, genetic factors, or beta amyloid peptide misfolded. What happens? You're going to see activated microglial cells. This activation of these microglial cells will start right away to release, as all of us know, COX-2, INOS, and the other products like the inflammatory cytokines like TNF-alpha, of course, interleukin-1 beta, interleukin-6. All right, this will lead to acute inflammation. This acute inflammation either to be resolved or it will cause chronic inflammation leading to the disease of the neurodegenerative disease. Okay, this is a normal condition that all of us are aware of. So now we would like to go and to know what happens in case of Alzheimer's disease. Alzheimer's disease, as we know, remember by memory loss, confusion, that leads to behavior and personality changes, i.e. decline in cognitive ability. It is the major type for senile dementia, all right? And the bad thing is incurable, degenerative, and terminal disease. So our purpose today, and I wish all of us can help each other, how can we stop such a disease? Or is there any way to cure? Of course, as you know, once it happens, there's no way to come back. But the only hope is just to early detect it so you stop progression. That's the first thing. Or you can do some protective measures so you can avoid acquiring that disease to anyone. And this is very nice. I just want to show you. You see, the third one is the second row. This is Alois Alzheimer, who detected the disease. And the first small one, this is Levy, that all us know that is this Levy bodies that happens, and it is forming a plaque over the neurons. Look what happens to the brain of this patient. They start to shrink. They start to be eaten. And now if you look at the hippocampus, as all of us know, of course, this hippocampus, it is a part or a major part of the limbic system. And the limbic system, this is the system responsible for motivation, emotion, behavior, and long-term memory. Look what happens. It shrunk. Once it is shrunk, and this is why we start to have this language problem, and we have to start to have memory loss. So it's a very bad disease, and it's devastating. Why? We have a neuroinflammatory reaction followed by oxidative stress. So what is the problem now? The problem now that link between neuronal damage and brain inflammatory process mediated by progressive activation of astrocyte. That's it. Okay? Leading to the overproduction, of course, of the pro-inflammatory agent. Misfolded, look at this one, misfolded and aggregated proteins. This means that, okay, when we talk about neurodegenerative disease, my dear friends, all of us know that there is a special protein that became misfolded, i.e. it cannot be eliminated by the ubiquitin proteasome pathway. And that's why it is precipitated over the neurons, causing the neurodegeneration. Not only that, because it became misfolded, it activates the microglial cells, leading to the strong immunological response, and they start to eat the brain tissue. Look now here between the normal and the memory, what happens, and to the language center. Okay, I'm sorry because I have to go a little bit more faster because I don't want to keep you that long. Okay, forget about that. Okay. Major problem now, you said that you have an immunological response, and not only that, you start to see a peripheral reaction. And this means that there is a breakdown in the blood-brain barrier. Breakdown in the blood-brain barrier. And this is why you can see this plaque formed from peripheral, and they start to go and precipitate it in the brain. Some evidence conclude disruption of the blood-brain barrier. Okay? And now, here is the new story that I would like to talk to you about it. Who can feel such changes? There is a very unique and special protein in our cell system. This is called APE1, which is apyogenic, apyrimidinic endonuclease 1. This protein is fascinating. It has like a dual function. Okay? First of all, it is like a DNA repair enzyme, okay, for base excision repair. And not only that, it is a redox factor. So once you have any change in the oxidative stress in the cell, right away, AP1 will be stimulated. And this stimulation, you will see the activation of the production of the other enzymes such as INOS, COX-2, and so on. It's a very amazing protein. I wish I can go through it, but you see, I wish we can share this information all together because I am sure it is very important in all neurodegenerative disease. I'm not sure about schizophrenia. yet. But you see, it is very efficient and very important. So what I'm saying is that once you have a problem, you see, it acts like an alert system. Puck. So this is what, like, it's a lighthouse. So this is AP1 puck. It is very hard. I'm going to show you now in the result in the model. We did our experiment hoping to stop the disease. So we did it in two different ways. We have in vitro study and we have in vivo study. In case in vitro study, you see that we use that PC12 used to study aluminum chloride as an inducer of the Alzheimer's disease associated with molecular alteration. In the in vivo study, we use Wistar rat used to investigate the molecular alteration also with aluminum chloride exposure and the possible therapeutic effect of two compounds, resveratrol and curcumin combination. So now we'll see now it's a combination. This means that can anyone alone can work? This is I'm going to show you right now. So now we have, remember, we have two keys. We have the key, most important key is the AP1 and this combination. We use cell line derived from so and so and so, as you know, and you culture the cell, okay? And then we're stimulated with aluminum chloride for 6 and 24 hours. The optimum sublethal concentration of aluminum chloride that induced oxidative stress was 0.5 millimolar. PC12 were treated with aluminum chloride and treated with either resveratrol, curcumin, or combination. To make the long story short, as you're going to see, okay, let me show you first step by step. I'm just watching just for the time. That's it. Okay, of course.
You have 3 more minutes.
I'm sorry?
You have 3 more minutes.
Oh, please be more generous.
3 minutes.
Make it 5 minutes. Okay. So now, as you will notice, all right, of course, the pro-inflammatory cytokines will increase. Of course, the key player, tumor necrosis factor alpha. All right, and you will see that it goes down when you treat it with curcumin, but when you treat it with resveratrol alone, it has no or little effect. But the combination was very strong. I hope you can see it quite well. All right, okay. And now you will see here how AP1 is affected. Once you induce it with aluminum chloride, right away you will see that AP1 will go very high. As I told you, it is a sensor, very important sensor. And not only that, guess what it's doing? You want to tell me, so what is the importance that it is an alert system? Yes, because it will stimulate the other transcription factors. It will stimulate the nuclear factor kappa B, leading to the induction of COX-2 and INOS, and also the AP1 enzyme, the transcription factor, I'm sorry. And this is how it is exaggerate the whole situation. And now, as you can see here, all right, you see how once we treated with the resveratrol and one we treated with curcumin, but the combination was very efficient to drop them down. And also here, all right, you see another point. I wish I can have time, but it's very clear that we don't have enough time. Don't ever forget the role of microRNA, my dear, because they are playing a very important role, because it could be a very strong target for elimination or to stop the reaction. So in this condition, you will notice that here that we measured LET7 family, all right, and you see they are very induced in the condition, and once it is treated with aluminum chloride, it goes down. Once it goes down, okay, look now, when I treat it with resveratrol, no effect. When I use it with curcumin, it goes a little bit down. But when I use both of them, it reverts them to the normal concentration of LET7 family. All right, this is beautiful reaction. We just did it like almost like 2 weeks ago. We'll do it in Bordeaux. You see, we would like to know exactly, just to make sure who induces or who affects the AP1. If I use the curcumin alone, to make it much easier, if I use the curcumin alone, all right, you will get very little effect. Of course, resveratrol has no effect, but the combination is very strong. And you will see, as you notice, as you see here, right, you see that the microRNA, the normal one, goes up, all right? And also here, as you notice, that the COX-2 goes down. Okay, I'm wrapping up. I notice. So in the result, okay, I think now it's my idea is very clear that when I use this combination of curcumin and resveratrol, it is really efficient, okay, and it really helps to revert this immunological strong interaction, and you can stop it, all right, and especially when I use curcumin. And I want to tell you something from my personal experience. I use curcumin. By the way, you should ask me, let me ask a question on your behalf. As you know that curcumin, it is not that easy bioavailable. The bioavailability of curcumin is very low. What should we do? We always buy it with bioprine. If you don't use bioprine, you won't see these reactions. Because you see, when I use curcumin with bioprine, this increases the bioavailability of curcumin. And of course, you can buy it even from special commercial companies such as Vitacost and so on. So this is very important. Don't forget that curcumin alone does not work. Okay. Well, it is the same story in the in vivo study that you see, as I told you, if you notice the effect on the COX-2 and the microRNA and how they are regulated by the combination of curcumin and resveratrol. All right? And of course here I'm sorry, this is COX-2. It is not COX-1, of course, on the diagram. I'm sorry about that. All right? But you see, you will notice that right away, once you have this combination, I was saying something and I forgot to continue. You cannot imagine how curcumin is efficient when you use it even in cancer patients, but not in the later stage. Curcumin is very efficient. It's like an amazing herbs. That's what I want to say about that, and I would like to finish my things. And as I said, this is my conclusion, as I said, and it was very clear that how this combination was very efficient. Okay, and attenuated aluminum chloride induced APO1 induction significantly, clears that curcumin function by different mechanism from resveratrol, as shown by alteration in APO1 and P53 level, right, as I show you in the result. PC12 work confirmed our animal study and highlighted the potential neuroprotective effect of resveratrol curcumin. As I said, there is no way to revert the condition, but the only hope that we can stop the progression, or maybe if someone is alert enough, if you can use regular curcumin, I'm sure it's going to be very beneficial for all of us. At the end, I would like just to acknowledge my group, Dr. Amira Zaki, and my PhD student, Mona Ataya, Ibtisam, Muhammad Abdel Qadir Adham, Asma Muhammad, and the collaboration with Professor Detlef Gabel from Bremen University in Germany, Professor Marc Landry from University of Bordeaux, Professor Jean-Luc Cattel, he is the leader in APE1 research from Ordine University. And thank you very much.